Hi, my name is Jessica Redline, and I'm going to be going through the lab 2 for this week. It is July 14th, 2020, and this is the first lab for Microbiology 225, and it's going to be aseptic and cultures. I'm going to demonstrate. I do have the instructions here just to double check and make sure that I don't miss anything, but I did prepare beforehand. I have my vials labeled already with permanent marker, depending on which culture I'm going to be doing. I also have like my alcohol cup labeled and ready to go. I already sterilized and labeled my pipette. So I already sterilized it with the alcohol, but I'm going to do it once again before I actually use the pipette, just to make sure. I also have my yeast going here. I did this beforehand. It said to dissolve about a half teaspoon in warm water and let sit for 10 minutes. So I did that beforehand, and I'm just swirling around waiting for it to get a little bit more foamy. As it said, I already have all my equipment on. I provided my own mask. I have my goggles. I'm wearing long sleeves, closed shoes. I already disinfected my area, and I also have 10% bleach nearby in case of any accidents or spills, and also for after. So first, I'm going to start with the E. coli test tube. It's instructed to run the tip of the tube through the flame just to sterilize it. So I'm just going to do that really quick. I have my candle lit over here. And then I'm going to set that in my little stand. I'm also going to repeat the same thing for the culture for the E. coli. This is the culture. So I'm just going to pass it through really quick. Just like so. So that one's done. And then the next step is, it says to pipe at 0.25 mLs of broth from the nutrient tube culture vial. From the broth tube, we're going to take 0.25 and we're going to put it into the culture tube. So I'm going to set my culture tube down, and I'm going to get my pipette, and this is labeled. So I'm going to go and get kind of hard because I don't want to get like air bubbles. So I'm gonna okay. So I did that. I put about 2.5 mLs of broth into the nutrient tube. A broth into the nutrient tube. It says to be careful not to touch this little thing. I don't remember the name of this, but this little pipette with the tubes, because you don't want to contaminate it. Remember, we just sterilized it with the flame. And so it says to replace the lid onto the culture vial and shake vigorously until the tablet dissolves. I'm just going to shake it until it dissolves. And then it says the next step is to, once this is dissolved, you're going to get the pipette again. You're going to draw the liquid from this culture tube and put it into the E. coli broth tube. Okay, I think that should be good. I think. Oh, I don't know if it's dissolved or not. I don't see that it's dissolved yet. So I'm just going to shake it a little bit more. It says to shake vigorously, so I don't know if I'm not shaking it enough, but I'm just going to keep trying and shaking it. I can still feel like the little tablet inside. Okay, that's good. It's dissolved now. So then I'm going to get my pipette and I'm going to draw this liquid from the E. coli culture. And I'm going to drop it into my vial without touching the edges because I don't want to contaminate. And so you can already kind of see, I don't know if it shows up on camera, but it's like a little bit, looks like chunks dissolve. And then I'm going to run the top of the tube through the flame again just to sterilize it before I cap it. Oh, I have the wrong cap. Okay, so I'm going to cap it. Okay, so we sterilized it. So I'm going to set this broth aside. I'm going to place my pipette back into my plastic cup that contains alcohol. I'm going to set that one aside. I'm just going to set my pipette back in my alcohol. It says to like run the alcohol through the pipette by drawing it and then releasing it to sterilize it. And then, so now we're going to do the epidermidis. So this is my vial right here that's already labeled. I am going to get my culture ready and have it next to me ready to go. I'm going to uncap this one, run it through the flame to sterilize it really quick. I'm also going to sterilize the culture vial really quick, just to make sure it doesn't get contaminated. I'm going to get my pipette that I've already sterilized with the alcohol, and I'm going to draw up 0.25 approximately of the broth into the culture. Then I'm going to cap it and shake vigorously. So just until I don't feel like the tablet anymore, it should be good. Okay, I'm gonna take off the cap and I'm gonna get my pipette again. I'm gonna draw the liquid from the culture and drop it into my test tube. And then I'm going to place this cap back on. I'm going to run this broth tube of the S. epidermidis through the flame just to sterilize it before I cap it. Okay, that one's done. And then the last one, I'm already used the permanent marker and labeled the yeast in warm water, about a half a teaspoon. I have my cup here, and I've already let it sit for approximately 10 minutes. I have my broth tube for the S. cerevisiae, if I'm saying it correctly, hopefully. And I'm going to repeat the same steps. I'm going to pipe it. I'm going to sterilize the tubes first. So just run it through the flame. And I'm going to sterilize my pipette into the alcohol. And I'm going to pipe it approximately 0.25. And drop it. Into my test tube. And that one's done. I'm going to cap it. This one's interesting because you can see it floating. It almost looks like when you get blood drawn and like the plasma is sitting separate from the blood. I don't know. But so all my 3 test tubes are done. I have it standing in this like tube stand right here. Makes it easier for me since it is glass. So yeah, and now I'm just gonna pour the remaining alcohol from the cup into the sink. It says that you can reuse and dry this cup after for further labs. And I'm gonna place all the empty culture vials, the E. coli one and the epidermidis, the staph epidermidis one. It says to put these vials with lids in plastic cup containing the yeast solution in a bucket of the bleach, undiluted bleach, for 30 minutes. And then the items may be removed and disposed in the garbage properly. And then after, obviously, I'm going to wipe down my area with 10% bleach once I'm done cleaning up all my stuff. And I am going to place these tubes into their designated area to incubate, and so that we can see what growth it has. I'm going to set them up in my cabinet in my kitchen where it's cool, dark, and it's protected from any drafts. So it's going to be my cabinet above my microwave, and it's away, from direct sunlight, and obviously my kids can't reach it, pets can't get to it, so that's the safest. And I'm going to incubate these for about 48 hours. After 48 hours, it says to inspect them and see if there's any microbe growth. And it says you can do that by holding up to light source. If not, then you could further incubate and see if there's any possible growth. But yeah, that is the lab experiment for this week, and I will upload the next video on the results of this. Thank you so much for watching.